I'll be telling you about our SPARC clinical trial investigating the sigma-2 receptor modulator CT1812 with a focus on our CSF proteomics biomarker findings. The SPARC clinical trial was conducted as a single-site trial at Yale School of Medicine to assess the effects of CT1812 on safety and synaptic density in a mild to moderate Alzheimer's patient population. What we did in this study, we took SPARC CSF samples at end of study at 6 months and at baseline. And we looked at differential expression analysis. And what you see here in the volcano plot on the left is that 128 proteins were found to be upregulated in patients given CT1812 compared to placebo, and 198 appeared to be downregulated. I'd like to point out that some of the proteins we've already looked at, including SMOC1 and YPL40, were significantly downregulated. Another notable biomarker I'd like to point out is clusterin. Clusterin is an AD genetic risk factor, also known as ApoE-J. So here we identified pharmacodynamic biomarkers of CT1812 that may reflect pathway engagement or disease modification. 14 of these biomarkers are previously well-characterized AD-related priority biomarkers, and as well as AD genetic risk factors. Next, we looked at pathway analysis, and what you see here are the top statistically significant pathways altered or that were differentially regulated in patients given CT1812 compared to placebo. And what you see here, the pathways are statistically altered, including inflammation and amyloid biology. We also noted in this analysis that many of these pathways overlapped with previous clinical trials studying CT1812 in a similar patient population. So we next asked, how might CT1812 be affecting the brain proteome of Alzheimer's patients? And for this, we leveraged with our collaborators a rich publicly available data set where brain and CSF proteomes were analyzed. So we mapped our proteins that were differentially abundant in patients given CT1812 compared to placebo to this brain network. And what you see is that several modules were represented, and of the top 5, 2 of them were synaptic in nature. And going back to our mechanism of action, this is very interesting because this supports, again, a synaptic protective mechanism of CT1812. Next, we looked at CT1812 ability to normalize biomarkers that were dysregulated in Alzheimer's disease. So what you see here, if we just look at the top example, you see that this protein is, and with AD versus control, is going up, and that with CT1812 versus placebo in blue, you see that it is going down statistically significantly. The priority biomarkers I mentioned earlier are in yellow, and you see here that the proteins are involved in key pathways disrupted in Alzheimer's, including autophagy, inflammation, and synaptic function. In conclusion, pharmacodynamic biomarkers of CT1812 were identified, including several which may reflect disease modification. Some of these are known to be genetic risk factors, and others are known to be involved in key pathways dysregulated in Alzheimer's, including autophagy, inflammation, and synaptic function. And lastly, this supports, again, a synaptic protective mechanism of action for CT1812 for Alzheimer's disease.